Hello everyone and welcome to this Qlucore webinar about analyzing RNA sequence raw count data. And we're going to use the new Qlucore Omics Explorer 3.10 product for this. For those that are new to the product, I hope this will give you a first feeling for how you can work with RNA sequence data in the product. And then, of course, you can use the product also for a number of other types of data. But today we're going to focus on RNA sequence raw count data and analysis of that type of data. So I hope you can see the presentation on your screen at the moment. On our website we have a number of case studies and other information that could be useful, but we have also put the information there about a new release 3.10. You're most welcome to visit the website and learn more about the product there. And as you know, Qlucore Omics Explorer is a fast and easy to use tool to analyze and explore data. You don't have to be a statistical expert. Today we're going to focus on RNA sequence data. And it could be both raw count data that you have or normalized data. And as you can see on this presentation here, you can work with many other types of data like single cell data, methylation data, proteomics data, etc. Some file formats you can directly import, like aligned BAM files, and you then need to have a GTF file as well. And you can also import quotient SF files if you have your RNA sequence data in that format. And we are going to have a look at the wizard, how you can import a matrix of raw count data into the tool and do analysis of it. And as you can see, there are also a number of other file formats that you can work with. In the presentation today, I'm going to use a raw count data set that's available as a gene expression omnibus data set. And it's also available in Qlucore Omics Explorer as a help data set. I'm going to import it manually, but it's also available under example files. So that's a data set that we're going to use for demonstration today. It contains a number of annotations also. So we got, for instance, disease state that we're going to use, and we also got information about gender, etc. For the variables, we have got identifiers like ensemble identifier, chromosome, gene symbol, etc. So all that information is available for this data set. The dataset that we're going to import will be imported using the wizard, and I'm going to show that to you in the demonstration a little bit later today. And of course you can also import BAM files, and in both those two cases you have the possibility then to do a normalization of your data after the import. So that's new in 3.10. You select the normalization in the data tab in the product, and that's what you can see here in the middle where it says input count. So it's count data independent if you have imported it through the wizard or if it's BAM files. And then there is a part where we got normalize, where you can select which normalization to use. And that could be dependent on also the type of test that you would like to do. So we're coming back to that a bit later. Here you can also select identifier. In this case we have got ensemble identifier, but if you also have got gene symbol you can select that, and you can also change in between them. In the wizard there is a question in one of the steps where it says, is this a raw count matrix? And that's where you then tell the system that is raw count data that you have in your data set that you are going to import. So if this is integer values that are raw counts, then just select the ready button yes here, and then you will get the data imported. And then you can select, once you have imported the data, the type of normalization that you would like to use. In the data tab you can also select some other things here. As I said before, it's possible to select the normalization type, but you can also select if you would like to change identifiers, if you would like to add pre-filters. If you have noisy data, it could be an idea also to filter your data to remove certain variables that are perhaps not expressed in your data set, and that's something that you can do here using pre-filtering. The standard normalization that you get if you don't do a selection is TMM. We will use VST, which is transformation or normalization that's used when you would like to do DSEQ2 analysis. But you can select here in this part which normalization to use, and for some of the normalization methods and so on, there is an option button here where you can add additional information to the normalization. Note that this normalization is then for visualization purposes. If we look at statistics, well, there are several different types of statistics. We call it statistics mode. Standard statistics, where you have, for instance, ANOVA, t-test, regression tests, etc. And then there is now, as of 3.10, also DSEQ2 test. And in those cases, you traditionally work with VST normalization. So if you know that you're going to use DSEQ2 for statistical test, then you select VST as the normalization. And then if you're going to work with standard statistics, then you can select TMM, TPM or FPKM normalization of your data. There is also extended statistics like before. That means that you can call R methods that have got wrappers from inside the tool. So that's also possible. In this example, a bit later on, we'll use the DSEQ2 statistics. And here we have got a number of tests. We have got likelihood ratio tests and Wald tests that you can select in between. They are a bit different. In LRT, likelihood ratio test, you compare two competing hypotheses, the null and the alternative hypothesis, and then you use the quantification that you get from the test to give you information about which of those that are the best supported in your data. You get P and Q values calculated as for any other tests. There is also another test that just use one model. It's the Wald test, and that's often called for when you're using just testing between two different groups here. But both for Wald test and for likelihood ratio test, there are also regression tests that you can select. For both tests, you can work with eliminated factors, and that's something that you can use when you would like to remove the influence of covariates for a test. And you can also work with restrictions when you just would like to test a certain part of your data set. You select that using annotations. You can select also to use independent filtering. That's often used when you do a DSEQ2 test. It improves the Q values, and it works in a manner where you then remove genes that have a low average expression to maximize the number of Q values that are below a given false discovery rate threshold. So that's something that we have as a default for the test, but you can select to change it or not to have it as a part of the test. And both fold change and difference are calculated for two group tests. If we look at the user interface, you select the mode here in the statistics window, and here you got all the possible Wald and LRT tests available here. And it's just in the same manner as if you select standard statistics, you then also get a drop down for those tests that are available there for standard statistics. So just select mode first, and then you select either DSEQ2 or standard statistics. Or if you would like to work with extended statistics, you can select that as well. As I said before, to visualize the data, you can then select a VST normalization, selected here in the Data tab, and there are options to that also. You can take into account your experimental design. So if you know, for instance, that you have some of your annotations that will be important, perhaps those one that you expect to see differences between groups, then you can set those up in options as well, and then use that as input to the normalization procedure. If you don't do that, you can use something called VST blind, where we don't take that into account at all. In the data export, you can either select to export original data that you have or the process data. In this case, we can see it's been normalized with VST. So it's possible to export either original data, raw counts, or normalized data. And the implementation that we have here, it's based on the article that we see here below. And it's independent of the original implementation in R. It's a much faster implementation. It's meant to be used in an interactive way. So we have made it much faster than you can do it in R. It's implemented in C++ instead, which is a much faster code and therefore useful when it comes to having it implemented in an interactive tool like Qlucore Omics Explorer. Here we have the data set. We can see here that we have some parts that are orange. That's the COVID-19 patients here. And we have one convalescent, green here, and the other ones are healthy. And we can see here that we're also using the gender annotations here with symbols to indicate which ones are females and which ones are males here in this data set. So let's see if we can run an example, and I will first start by importing the data, and then we'll do the analysis after that. So let's start with an import. As with Qlucore Omics Explorer, most of the importers are available under File here, where you can import both BAM files, and you can also import raw count data in a matrix, perhaps a tab-separated text file or a CSV file. So I will start with Open with Wizard, and then I will take this COVID dataset here and import it. First step here, just an information about the wizard. Then the next step here, this is where you select if it's raw count data or not. In this case, it is raw count data, so I changed the radio button from no to yes here, indicating that I have raw count data that I'm going to import. Then I press next here. Separator is tab, so I don't have to do anything. Press next. Matrix orientation, variables as rows. Just press next. Data matrix, here we selected top left corner of the data, which is this cell here. So I just click there, press next, and then I go to the last sample here, but the first variable, click here to take that information as the matrix to be calculated on. Press next. And then I can bring all the variable annotation into the tool. You can see we have both ensemble identifier and gene symbols and so on. Press next, and then I take the corresponding information about our samples. Here we have disease state and gender, etc. Press finish. You can see that the data is read into the tool, imported, and then we can see it here in a PCA plot directly. If we go to the samples tab here, we have all the annotations, and I'm going to use disease state as I talked about before. It has got 3 groups. Most of those are in the COVID and healthy groups here. I'm going to color according to that, and we can see in the PCA plot the 2 different groups here. If we look on the top here, we can see the data tab has been opened up, indicating we have count data. We can select if we would like to use ensemble identifier as the identifier for the variables. I will change that to gene symbol in this case here, and I will collapse the data. You can use geometric mean or median or something like that, select median in this case here, and then the data will be recalculated, of course, since we're changing the identifier for that. Here we have the normalization, and here we can see that TMM has been selected by default, but you can also see that you can select other types of normalization, so FPKM, TPM, and VST, and you can change some of those. For instance, TMM, you can change the length to something else here using TMM reference counts and so on to do a better calculation. I will also here in this data set, I will use another annotation called gender here. I got females and males and use that as input here so we can see which one is which here. Males are currently squares, no, females are squares, males are boxes. If I would like to change one, I can change from cube to, for instance, octahedron or something like that, and you can see that the plot is updated accordingly. In the statistics, you can select if you would like to run a standard statistic test, which is available here. Then you have the test down here. It's the traditional one, t-test, Welshman, Whitney, ANOVA, cross-call values, etc. In this case, I'm interested in running a DSEQ2 test, and then I select mode, DSEQ2, and then I got the possibility to run that. And I can set up here also which type of test I would like to do. You can see that they are available in a drop-down list here. We've got the Wald tests, two-group, linear regression and ranked regression, and also paired two-group. For likelihood ratio test, we also got multi-group and two-group, regression test and paired two-group tests. I will select the two-group for Wald here, but we could have selected something else, of course. And then I will use disease state, and I will take COVID against those that are healthy. I will skip the convalescent, that's just one sample here. But otherwise, you can, of course, use that, or you can omit it like I did in this case in the test here. You can add restrictions if you just would like to test, for instance, on the males. If you would like to use the gender information as an eliminated factor, you can simply say that, yeah, I would like to use that and remove the influence of gender when I do a test. It's just a two group because we just got males and females, as you know here. And then you can press calculate to do the calculation here. Before that, I think I will also select here VST, and here you can select in the options tab here if you would like to use one of the factors to estimate the when you do the calculation of the dispersion. So you can use one of the factors like, for instance, disease state and so on for that. But I will use the blind one here. So I just press calculate, and then we get a new normalization of the data. And when I press calculate here in the statistics, we are going to calculate the P and Q values and so on. There is a calculation button just because there are so many different things to set up and change. So we have selected to have a calculation button there before you can use the sliders. So first it's calculating the values here, so we can then filter on P and Q values and filter by fold change also. So all that information is calculated, and then you get a presentation of the data. So now we have both VST normalization of the presented data, and you can see the sliders are now also active. So now I can either use the sliders like this. You can see how the PCA plot is updated accordingly. And you can also, as before, just set the fixed value here. So if you would like to do that, you can set the value, and then the sliders are updated accordingly. Here you select if you would like to use independent filtering, which is often recommended when you have a data set with raw count data. And you can also select which value you would like to use for the false discovery rate here. We use a default value of 0.1, which is often used. So all can be changed here. It's just to uncheck here, and then you can use another value for the FDR, or uncheck not to use independent filtering. You can also filter by fold change, as you normally do as well, and you can then use the slider for that as well. So sliders are active once you have pressed calculate and so on. If you do changes and so on, you press calculate again, and then sliders will be active after that. Now, if we look at the results and so on, you have that as always in the variables tab here. So we started here with 60,000 variables. We are now down at around 800 variables, the best discriminating ones. We got them as gene symbols here. We can then, if we would like to add information about, for instance, P and Q values here, and also fold change or difference values. Difference, that's the log fold change values. And you can order them just by clicking on the column headers like this. So you get them from the lowest to the highest value and so forth. So all that information is then available here. If I would like to save the information here, as before, you just click on something, make a copy like this. Discriminating variables, for instance, and if you would like to export it, you have the icons here to import and export. If I select export here, you can see that you can include your annotations and data, and here you can select then if you would like to have your process data, original data, and that will depend on the changes that you have done here. For instance, in the collapse and so on, I've changed the data there, so I don't have the original there. But when we do work with DSEQ2 analysis, remember all the statistics and so on, that's made on the raw count data, while we in the visualization and so on work with a normalization presentation that you see here, for instance, in the PCA plot or in other plots that we have. And then just press OK, and the data is then updated in a variable list that you have externally. If you would like to save your data set and so on, you go to File and Save As, and then you can save your data set. That's recommended after an import also. And then you will have your data set as a GData file on your computer. So that's the same procedure as we normally have. And here, as you could see, we could filter down to quite a low number here. So it's a good discrimination in between those that are COVID patients and those that are healthy in this data set here. If I would like to present it in some other way, it's just like every presentation. You set up new synchronized plots, and then you can tile them so that you have them side by side, so they are synchronized here. If I would like to present this another way, I can do so. For instance, you can select here a heat map if you would like to have that. You can also select here UMaps or TSNIs or whatever. So if I select UMap here, just need to do calculation here with the calculation button, and then the UMap will be presented here in the synchronized plot. So there is a multitude of plots that you can select in between, and some of those of course more suitable when you have a large data set like heat maps, UMap plots, PCA plots, TSNI plots, etc. So either you select to have it in a plot like this or something else, and here of course you can then you do the same as we did here. We can color according to the annotations. So if I select here now disease state here, I can color according to that here as well, so we can see which one is which. And if I would like to use one of the other ones like gender and so on, I can use that and then use that information here as well, just in the same manner as we have it in the PCA plot here. So it's a lot of possibilities here that you can work with in both the PCA plot or in the UMap plot and so on. All information about this is available with the new functionality under Help here. So if I go to Help and Documentation and Help Manager, you will find this. You can go to Search here, and then we can take DSEQ2, for instance. Press Search, and then you can read more about DSEC2 tests and so on, about the framework and so on that we have implemented. If you would like to learn more about VST normalization and other normalization methods, you can just write that, and then you get also the information about how they're calculated, etc. So this could be a nice information that's available there. Now, this is one way of importing data using the raw count data in a matrix that you import using the wizard. There are, of course, other ways, and I mentioned BAM files. So if I go to, for instance, File here, there is also Open BAM files. And then you need to specify your BAM files that you would like to import, and also corresponding gene transfer file. So I will just take an example here. I will use some of the files that I have available. We'll use that in our courses folder, I think. Let me see, short webinars, and I will take RNA-sequence data, BAM files, take one of those, and then I will select the number of files here also. Let me see, and the BAM, I've got a few of those, like this. So you select your files or a folder, and then the GTF file, you press OK. Data is imported, and you will get a PCA plot, just like for the other ones. So we have the plots associated with the COVID data here, and then we've got the 4 samples here that have been imported as BAM files. And also for this, you can select which type of normalization, of course, and select that as well. And you can set up a similar statistics test. Now for BAM files, you often don't have the annotations available, so that's something that you often need to add. So if you have your annotations, you can add new ones. I will then, in this case, go to Import here and select Sample Annotations. I prepared 2 groups for this dataset here, and then the 2 groups will be imported as well. It's just 4 samples in 2 groups, and then I can color according to that. So we see that we have 2 groups, 2 gray samples and 2 red ones, and setting up a test is the same procedure as we had for the COVID dataset. You just select which test you would like to run. I can select DSEC2 here as well. 2 group, perhaps a Wald test this time. No, LRT test, sorry, and then I select annotation, groups here, and then I select group 1 against group 2, for example, and then I press calculate here. Calculation is done, and then I have done the possibility to use the sliders. If I would like to change the presentation to VST, I can do that, and then I press calculate to do that, and then calculate here as well, and then we have a presentation with VST, and then we can use the slider in the same manner as we did for the COVID dataset. So BAM files, that's an alternative to import your data. If you have aligned BAM files, a GTF file, you can just import your data. If you have it as a count matrix, you use the wizard. There is also a third possibility. If you have quant SF data, then you can also import that. That's also raw count data. So that's a possibility as well. And quant SF files are normally from Salmon libraries or from some Illumina machines that create data in that format. At the end here, I just would like to mention also that if you work with a new functionality, there are some information available in the videos and so on that you can have a look at. And there is also the help here that's available here with the new functionality, and the COVID dataset. If you would like to work with that, that's also available under example file. So you don't have to import it like I did, it's already here, and then you can play around with that dataset, which is a raw count dataset. You can directly work with it. But please, if you have questions and so on, you are most welcome to contact us at Qlucore with your questions, and I hope that you will find the new functionality useful for you. If you have questions about how to work with the new functionality for RNA sequence data in using DSEQ2, then you are most welcome to contact us also for more detailed information. The data that you have seen now will also be presented in another of the webinars that we have, so we'll use this data set also for other types of courses and for different exercises that are associated with those. If you have any questions here at the end, you can just